from Microbe TV, this is TWiP, This Week in Parasitism, episode 228, recorded on January 26, 2024. I'm Vincent Racaniello, and joining me tonight from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and everybody else. The weather outside is frightening, as the opening line to a
Why is it frightening?
Song.
Why? What's wrong?
No, the weather outside is frightening.
I have to get home tonight, you know?
Tomorrow you will see why tomorrow's weather is frightening.
All right.
But it'll be too late by then, won't it?
Well, I need to just get home because it's always difficult.
Forearmed is forearmed.
Forewarned.
Forewarned is forearmed.
Also joining us from New York, Daniel Griffin.
Hello, everyone. And, you know, I waited my turn, but I believe it's frightful.
Frightful.
Frightful? No,
Yeah.
But it doesn't rhyme with lightning.
Well, but I think it's, and the inside, the fire is delightful. So it's frightful
that is
and
all
delightful.
of the above. All of the above is
But
true.
yeah,
That's true.
I actually thought it was quite nice out. I usually try to avoid talking about the weather, but I got out for a little bit with the dogs today, and it's actually warmed up a little bit here. So.
Oh, warm. I just got back from Iceland, so I think everything is warm compared to there. But it's true. I was there just 4 days. We went whale watching. The sun was out. They have 4 hours of sunlight during this time of the year, and it's on a ship just in the harbor. It didn't even go very far. The guy says there's a whale, and there was a minke whale.
Wow. And
everybody rushes up to the deck. Right? There must have been maybe 2 knots of wind. Maybe a little bit less than that. The bone chill factor was like no one stayed on board the ship. They all came back inside right away. Okay, I've seen the whale. Okay, now let's go.
Yeah.
That sort of thing. But it was really, 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 really cold.
Also joining us from Glasgow, Scotland, Christina Naula.
Hello, everybody. I haven't been anywhere exciting, just in my back office. But it's great to be here this evening.
Yeah, I could have actually yelled across from Iceland to you. You were right up next door.
Yep, it's just a stone's throw.
Daniel,
have you ever been to Iceland?
I've not been, no.
Daniel, your daughter was here the other night, and she said that the studio looks a little fancier than what you have at home.
Apparently, it is fancier there at the incubator than it is here in my.
But you do have a light, right?
I have a light.
Yeah, because I see it's bright.
Yeah, no,
light that's bright.
It is bright. Actually, my daughter was in last night because she and I were headed to a Burns Night celebration. So when I saw Christina today, my first question was, have you recovered? And I think she had missed out. But, we did the piping in of the haggis with the bagpipes and the reciting of the poems and finished the evening with the crossing of the arms and the singing of the Old Lang Syne. So,
did you wear your tartan?
Yeah.
So it was a mix, right? So it was a you could either come in black tie or your tartan. And being Irish, we don't actually have a family plaid, I guess, or tartan. So I went with the boring black tie, but yeah, there was a mix. There was a mix of folks.
You have a good time?
It was a good time. Yeah, it was a good crowd, and there's I think it's sort of a fun crowd comes out for Rabbi Burns Night.
Who does the readings?
It was around the table, right? So there was
really?
Yeah, different
nice.
people read at different times, and it was sort of funny. I was talking to I sat next to this woman Morgan and her husband Malcolm, and we're talking for quite a while. And then they're like, "Oh, what Burns Night would be correct or full if we didn't take someone whose last name was Forbes and get him up to read a poem?" And that was actually Malcolm Forbes, who I was chatting with.
Get out of here.
Whose family has nicer boats than I have, by the way.
Oh, yeah, I'm sure. Nicer buildings also.
Who else famous was there? Can you remember?
Yeah, there were a number of it. It was, it was a
He doesn't want to drop
nice,
names. Okay.
nice fit. Yeah.
Justina, have you
just
done
hello
one
to
of...
Malcolm, who wants to come frostbiting with me, so he has bad judgment, just like the rest
Christina,
of us.
have you done a Forbes thing?
A Forbes thing?
No,
Burns.
No, I actually missed it out. I'm not very fond of the haggis, but my two younger
you're
boys...
from Switzerland, come on.
I know, I don't need to. I'm not bona fide Scot. But we do usually have Burns night, just in the house. But my boys went to a ceilidh dance last night. It's another tradition on Burns night.
I went to a...
Which is amazing.
I went to a ceilidh once at a meeting. And they were explaining it. It was in Glasgow, was it? Maybe, but we all danced around in a big circle, right?
It's so much fun.
In fact, I have a video of it on my YouTube channel. You can find it there.
I'm going to go and Google that.
Yeah.
In my high school, you had a woman who had just come from the northern part of Scotland, and she has a very thick accent, and she was asked to read the poems in their native tongue. Basically, they don't sound anything like English. It's just absolutely. Did you get all the words, by the way, Daniel, when they were reading them?
So
if there was something,
yeah. So some of it is written right in the Scots. So it's I need the prompt. There was a lady who was with us, and she had it memorized. Right, she was proper Scottish descent and had most of the poems memorized. But I need my cheat sheet.
Did they read the one about the wee mousie?
Yes. Yeah.
Oh, good.
Speaking of poems, did you know that Winston Churchill wrote one poem in his life, and it was about influenza?
Really? And it goes something like...
No, none of that stuff. No. That's not about the little girl who opened the window in influenza. That's something else. That's not Winston Churchill.
I'll have to check that out. That's interesting.
He writes about the 1918 influenza, yeah.
Wow.
He was quite a painter, by the way.
I heard. All right, if you enjoy our science programs like this one, we'd love to have your support to help them continue, because we don't have ads for any of these programs. We don't monetize. You go to microbe.tv/contribute, and you can make a donation in many ways. And it's in the U.S., it's federal tax deductible. So microbe.tv/contribute. Today we have two papers for you, and we'll start with the Fountain of Youth, the Parasitic Fountain of Youth from Christina.
Yes. So, I'm going to tell you a little bit about how tapeworms can extend the lifespan of an ant. It's not a new paper, but I came across it and I thought, this is really fun. I want to talk about it. So the title of the paper is Extreme Lifespan Extension in Tapeworm-Infected Ant Workers. The authors are Sarah Beros, Anna Lenhart, Enon Scharf, Matteo Negroni, Florian Menzel, and Suzanne Wojcik, and they hail from Germany and Israel. So it was published in 2021, so not very new, but, exciting. And it's published in the Royal Society Open Science Journal. So I'm going to get my notes open. So this paper tickled my interest because I just find it really fascinating that even the tiniest of organisms can be parasitized. To think that an ant can be infected with a tapeworm is really quite mind-boggling. Mm-hmm. Although, of course, the tapeworm is not the size of a tapeworm I have in my mind, in my kind of visual imagination. And actually in that paper, it's not the adult tapeworm either. We're going to be talking about the larval, the cysticercal stage, so the intermediate stage of the tapeworm. But, it's still mind-boggling. The ants are tiny. So these particular ants are about 2 millimeters in length. And I also find it really interesting how parasites can influence host behavior and also, change some phenotypic traits. And in the case of this research, actually lifespan. So I think it's been shown many times that parasite infections in social insects actually can induce changes in behavior. And, they can also accelerate behavioral maturation of young workers. So it's really quite interesting. And this paper looks at one aspect of how parasites of social insects can influence those ants. So, social insects they live in that kind of social environment, like in nests or in colonies, really. And these ants, they actually benefit from this social environment in regards of their health. For example, survival improves. And this is particularly apparent in queens who live for a very long time already. So I didn't know that actually a queen ant can live up to 10 years. And during that entire period of time, they remain fertile and they produce offspring. Essentially, the queens are very pampered. They are very well fed and they're very well cared for by the colony. And they're also deep inside the nest, and they're maybe not so much exposed to the dangers of the outside world, which no doubt probably it must contribute to their longevity.
They don't go shopping, do they?
I don't think so, no. But that lifestyle, so that kind of living together in close proximity, also makes them more vulnerable to parasites. They are genetically very similar, and they live in crowded conditions, so that makes them quite vulnerable. So usually parasite infections have a fitness cost to their hosts, maybe slower development or reduced survival or lower fecundity. And so that's why really that paper title came a bit as a surprise. So that some parasites can extend the lifespan of their hosts. Maybe the parasite reduces the reproductive success of the host or even renders them sterile, and that kind of affects the trade-off between fecundity and lifespan, because having offspring is expensive. So that's maybe one way.
Christina, could you just remind us as to what the definitive host for this tapeworm is?
I'm just coming to that, actually. So I was just ambling my way towards the paper.
Okay. I thought I'd missed something here, but no.
No. So the background for this paper is the finding that Central European ants of the species Temnothorax nylanderi live longer when they are infected with the intermediate stage of the tapeworm Anomotenia brevis. So the final host of this tapeworm is a woodpecker who eats the infected ants. Yeah.
Cool.
Yeah. So I haven't really looked up any more about the life cycle of the tapeworm because I just wanted to focus on what's going on in the ant. But maybe we can Google it and put a little note in our show notes. So just a quick word about the ant species, really, and the different types of ants. So for this particular ant, the colony size is about 280 ants, so that's not massive. Because in my mind, I kind of have these anthills of my childhood, and they were absolutely ginormous, but these ants actually build quite small colonies. They kind of live under kind of leaf litter and in little crevices. And actually the size of that also explains why the researchers were able to look at 57 colonies altogether. So most ants in general are female, and males are essentially flying sperm. And they're not really relevant at all in this paper. So there's one queen. Some ant colonies have multiple queens, but in this species there's one queen. And the rest of the colony is composed of female ants with different roles. So younger ants, they will be nurses of sorts. They stay in the colony and they tend to the needs of the queen and the eggs and the larvae and the pupae. And as they grow older, they become foragers. Or that's what they call them in this paper, and that involves essentially going out looking for food, but also defending the colony. So the paper often mentions foragers and nurses and workers, but I think actually what I'm going to do for the purpose of this snip is I'm just going to lump them together as workers. But I'm not an ant researcher, so I hope I'm not committing a grave crime here.
But you are an ant, not. I'm sure, right?
No, actually, am I? Oh, yeah, I am. Yeah, sorry. I hope none of my nieces and nephews listens. That would be embarrassing. So where was I? So before they started looking at that, at these long-lived ants, what already was known that the ants not only live longer, but they also have other traits that change when they are infected with that tapeworm. For example, they're less active, they stay on the brood pile, and they don't run away when they are threatened.
Yeah.
So,
Christina, the tapeworms are always in the ants? They're past?
So it's the cysticerci that are in the ants, and then when the woodpecker comes along and eats ants, presumably in a nest, so that they will be infected and they will exist somewhere in the intestinal tracts of the woodpecker and develop into the adult worm.
So in the ants, though, are they passed from ant to ant?
No, they just stay. So the ants get infected by ingesting the eggs, and
I see.
I actually forgot where they pick up the eggs.
Okay.
Presumably when they're foraging outside. So
and the woodpeckers are shedding the eggs, I presume, and that's the cycle. Okay.
Yeah. That's why I understand it to be. Actually, also, it was quite interesting that genes are differentially expressed in these ants, particularly in the brain, but also in the abdomen, but the paper doesn't really discuss that further. So the research question for the paper was how long infected worker ants can actually live, and maybe learn a little bit more about why they do have an extended lifespan. So they tracked worker and queen ant survival for a whole 3 years, and they looked both at infected and non-infected individual in a total of 57 colonies. And actually they marked the ants. Now I don't know how, I presume they must have artificially infected them, because otherwise I don't really know how you can tell an infected ant from a non-infected ant, but they I don't think they mentioned that in the paper. So, on the whole, it's not really clear what regulates the lifespan of ants, but they have shown in that study that, for example, the infected ants are fed more often than the uninfected ants. So maybe they got more social attention. Well, they did definitely get more social attention. And maybe, I'm just thinking, maybe a well-fed ant is a happy ant. In humans, well-nourished, wealthy people generally have a better life expectancy, but that's just speculation on my behalf. But they looked at why the infected ants actually received more social attention and care as well, apart from looking how old they got. So I'm not going to go into any detail of methods, but what they found is that infected workers survived considerably longer than uninfected workers. I'll give you some numbers in a moment.
By the way, Christina, they say that you can distinguish infected and uninfected by their color.
Oh, I missed that. Thank you.
Color morphology. So, because they say that we collected parasitized and non-parasitized colonies, so there's a way to distinguish them.
Oh, thank you. I completely missed that. I maybe did skim over the methods a little bit.
It's all right, no problem. Because I was wondering if they can't do PCR because you got to sacrifice the ant to do PCR.
Yeah. Well, I just kind of thought, well, maybe they artificially infected them and then just popped them in a colony. In the woods. But they did. Okay,
they can see them.
Thank you.
Yeah.
So you can see the rate of predation that the woodpecker inflicts on the colony, basically, right? Because all the ants there should live to about 800 days. And the curve for going from 100% down to, let's say, 50 is about 250 days. And then, but the woodpeckers are having at them. They're not using outdoor colonies. They're taking this inside and then studying them in a terrarium.
They're lab colonies.
In a terrarium. Okay, fine.
Yeah. They're lab colonies. Yeah.
Okay, fine.
Yeah. And they actually also feed them and water them and, look after them, so they provide the food that they need.
Gotcha.
Okay, so the infected workers, they live a much longer. And also, actually what happened is the presence of infected workers reduced the survival of their uninfected nest mates. And this was much more obvious in older workers than in younger workers. So that's interesting. And then uninfected workers in parasites colonies had I think I just said that actually. I'm just repeating myself. So they had increased mortality. And queens, which were not affected, survived for an equal length of time, whether or not the colony was parasitized. So the parasite didn't really impact the survival of the queen. And overall, the colony death, so all the queen and all the ants dying, was about the same in infected and non-infected colonies. And when we look at individual ants now, so they did mark them, and so they were looking at the actual survival rates of actual individual ants. So uninfected workers survived about 250, kind of somewhere between 250 and 300 days, but infected workers lived up for over 800 days. And there were considerable error bars, but, it's still impressive. I think that's like a good 3 times more, 4 times more. So essentially more than half of the infected workers were still alive after 3 years, but all of the uninfected workers had already died. So the 3 years was the study period. So I don't know, that's astonishing.
Did the behavior of the infected ants cause the uninfected ants to sort of take care of them rather than do their job, and maybe they burned out?
I'll try and answer that, Dixon. I hope you're going to be satisfied.
Oh, so whatever you say, I'll be satisfied.
Well, I don't know. I mean, there's still lots of questions. So we look at that in more detail, there's 2 lines of evidence. So the first line is the infected ants. They just seem to be aging more slowly. Now, I'm not going to take you through it because this is a snippet. I'm not going to talk you through the figures. But for example, looking at the ants' metabolic rate, it appears that older infected worker ants have similar metabolic profile to those of the youngest members of the colony. And usually when people age, they slow down. And that seems to be the case for ants as well, but not in those infected ones. They seem to have a similar body mass. And in the absence of a queen, what would normally happen is that some younger ants would then kind of develop ovaries and potentially turn into queens. Mm-hmm. And it looks like the older workers are actually infected workers are also able to invest energy into ovaries, which normally only the younger ones would do. And finally, they also behave more like younger nursing ants in that they stay close to the brood. Normally, older ants would go out and about foraging. So essentially, they seem to remain longer, or in other words, they age more slowly. So the aging process seems to be slowed down. And then the second line of evidence is that infected ants are looked after very well. They receive more social care than even the queen. And in a way that probably makes sense that would have an effect on their lifespan, because those, they were well cared for, and essentially the other ones were working their socks off, looking after the infected ants and all the offspring. Right. And that's maybe why they had increased mortality, but the paper didn't look into that. So we have a bit more information about why the infected ants attract more social care than the uninfected ones. So they're not fed more, or they don't get more attention because they beg or ask for more. And actually, to kind of look into that in more detail, they undertook an experiment to see if cuticular hydrocarbons, which must be some sort of chemical messenger, could explain why infected ants were better cared for. So in a nutshell, they extracted those hydrocarbons from both infected and non-infected ants. They coated glass beads with the extracts and then exposed uninfected ants to those in a Petri dish. Mm. And that showed that the uninfected ants showed significantly more attention to the glass beads that were coated with infected ant extracts. So that suggests that maybe the infected ant cuticular hydrocarbons are more attractive for ants, and this is maybe the reason why they got more attention. But how the infection actually causes the hydrocarbons to be different is unclear, and they haven't really looked at that yet. I guess that might be work in the future works. And So I'm just going to close just by giving some thoughts. So usually parasites or being infected by a parasite is costly for the host, but in that system the negative effect seems to be to the uninfected nestmates. So these had a really significantly increased mortality rate, even though the lab colonies were well looked after. So maybe it could be that the increased food demands of the infected worker that increases the workload of their nestmates, and that could then contribute to the mortality. So essentially, they just work themselves to death in not very scientific terms. But that's not supported by the data on metabolic rates. So the uninfected ants seem to be under no more physiological stress than their mates. So we don't really know. And then I was also thinking, well, what's in it for the tapeworm? There must be a reason. So the infection changes the behavior of ants, which I have mentioned. So infected ants stay inside, and apart from living extraordinary rate, the kind of infection reduces the behavioral repertoire of the workers and also the anti-predatory responses, which I think I mentioned earlier. And that's quite different from other helminth ant infection. So they mention a lancet liver fluke that infects ants, and those ants leave the nest to become more exposed to the definitive host of the parasites, which are larger herbivores. But if we think about the feeding habits of the final host here, of the Anomotinia brevis tapeworm, the woodpecker, I think the behavior change in temnothorax worker maybe makes sense. So they're tiny and they move around leaf litter, and they'll be difficult to spot for the woodpecker. And woodpeckers, they kind of like to search for larvae in stick nests and other nooks and crannies, which is exactly where those ants form their colonies. So keeping the ants in one place, a place where the woodpeckers look for food, is probably a really good strategy for the infection to become carried on, or for the ants to be found and eaten by the woodpecker. So that's really the kind of sum of it. So I don't know what you think. I'm really quite keen to learn more about parasites that infect tiny creatures and affect their behavior. So maybe, if you don't mind, in the future, if you're interested, we could do the occasional weird parasite snippet.
That's not so weird. This is a great study. I love it. So, are there other experiments that you thought of that they could have done to get more evidence that sick or shall we call them disabled ants require more attention and therefore place a heavier cost on the non-infected ants, and therefore their lifespan is shortened?
I don't think they're any more sick. They're just more lazy.
No, but I would love to see what happens if you infected a colony, and a proportion of the colony became positive for the parasite. And Vincent said that they change color so that it's easy to recognize them apart from the other ants, and then remove them from the colony. And see what happens to the colony after that. Yeah. And also, can an infected ant take care of itself? Yeah. And if not, then that's a cost.
So this is the unique structure of an ant colony is possibly mediating this effect, right? The fact that ants take care of each other.
Yeah.
Well, they do because their lives depend on it.
I'm saying that's maybe responsible for this enhancement of longevity.
So how much of a caste system is this species of ant? Because there are workers, and then there are defenders of the nest, so to speak, and then there are these other groups. The reason why I'm asking is that when I was a postdoc at the Rockefeller, someone was doing their PhD thesis on termites. Mm-hmm. And termites use a chemical caste system. The chemical is passed down from the female, from the queen, and then it's processed, and then it goes throughout the entire colony, and the colony differentiates into a series of chemically induced castes. including the workers that do all the work, of course. And not all the time, they're not always working. They just look that way. The same is true in bees. They've marked the bees, and they do a lot of resting, even though they think they are all workers. They're not as good of workers as you or I would be if someone were watching us. So this guy, he got his PhD at Rockefeller, and he got him. A lot of comments came through as to what is the basis for this chemical caste system that almost all colonial insects seem to have. And so I wondered what proportion of this colony were workers, what proportion were the soldiers, so to speak, the guardians. We have drone bees. Do we have drone ants that don't do anything? Who becomes infected?
Well, I did. I mean, it must. I don't know because I didn't look into it, but
okay.
I did mention that the males are essentially just sperm donors. And
right.
I did lump the kind of female workers all together, but they do actually look at foragers and nurses. But the kind of the foragers are classified as those that go out and find food, but they also defend the colony. But I didn't really go beyond any of that because I was mostly interested in what the worm does.
This comes out of Germany, correct?
Yeah, I think so, yeah.
So there's a wonderful book on ants written by E.O. Wilson and his partner in crime. His name was Holdobler, and he was in Germany. I forget where he was in Germany, but they produced a very, very thick book, which, Vincent, I think you have a copy of somewhere in the incubator. Some of the books that we brought from my part of the thing.
Maybe,
and E.O. Wilson actually signed the book.
Cool.
And you know how he signed it? E.O. And then instead of saying Wilson, he drew a little picture of an ant.
Yeah.
A little tiny ant. And that was his signature.
Christina, what human tapeworm would be the closest to these? And
well, I don't know. From the names, it has a tenia in the name, so I'm not sure. I didn't look at that either.
Cyclophilidium?
I just want to know if there's any evidence that tapeworm infections of humans make them live longer.
Well, it would be cysticerci, so the ants. So the ants are not infected with the final tapeworm, but
true.
with the intermediate stages. So I don't know.
So
I don't think there's any evidence in kind of people with cysticercosis that they live any longer.
Right.
So the ants just have eggs in them, right? And they just
they have to see they have the kind of when you have cysticercosis, you've got these little cysticerci, these larval.
That's what the ants
have.
So
that's what the ants have.
Okay, got it.
Yeah. Sorry.
So it's an aberrant host right? Because cysticerci in people aren't.
No.
Intermediate. It's an
intermediate host. Yeah.
No, it's a required host.
I thought that when you get cysticerci in your brain, it's because you ingest the wrong form, Dixon.
Yes, maybe I didn't use a good example in people. Okay. We are the aberrant people.
We are a dead end. We're a dead end.
But the herbivores would be...
Okay, got it.
They would be the intermediate host because they tend to get eaten by carnivores, whereas we don't. Not often.
As we were doing this, Dixon, I was waiting for you to talk about Yul Brynner. And maybe we'll get there, but maybe other people may not remember. But there's, a couple things I thought was interesting, right? One is, here they're suggesting that this infection, is beneficial, right, for the infected, but not necessarily sick. So sort of an interesting thing there. They're not necessarily sick. They're giving off these volatiles. Everyone's taking care of them.
It's a chemical message.
They get to stay home in the nest.
That's right.
Which maybe is not so great, as we find out, because what is your concern with it? How many ants get to live out their life? A lot of them get eaten. So, in the world, in the world, then maybe it's not so great to be tapeworm infected because then you're hanging out where the woodpeckers know to come.
Yeah.
So because sort of wondering what's the evolutionary advantage?
Maybe it's
what drives this.
Daniel, you hit on a wonderful point right now, because the infected ants might act as a beacon to the woodpeckers.
They're a different color. They're all clustered together. I
the
even
next thing
wonder
you
about
know-
the volatile. What does that volatile effect have on the woodpeckers finding them? Is it really
Exactly,
It's one thing to be
exactly.
between two sheets of glass in a lab and everyone's taking care of you, but
No, you're
you know,
right.
is this really an advantage in nature out there in the world? But they do. They mention they've got a bunch of references here, like half a dozen, where. This is not the first time that lifespan extension has been described in host parasite systems. So it's sort of interesting. Do you still call it host parasite if the quote unquote parasite is actually helping you?
Right.
Is it still really a parasite if it's something that actually benefits you? And that made me sort of think of the example with maybe trichinella might have some beneficial properties.
Well, it does actually. It would prefer. When I say prefer, they weren't that smart. They weren't that smart. But imagine now if one of your evolutionary, what am I trying to say? Characteristics that you've been selected for is to allow the host that you infect to be resistant to perhaps other agents which might shorten their lifespan, and not allow them to go further away from where the source was originally acquired. So that an animal that's a predator or a scavenger that eats a portion of a carcass and then goes, another 200 miles and then eats another carcass, and then finally it becomes a carcass. For instance, all of the grizzly bears in Yellowstone Park are infected with trichinella, all of them. And it's true also for a lot of peridomestic black bears. And so the further away from the original source of the infection that you can move those scavengers before they die, it expands the host range of the parasite. Trichinella doesn't have a specificity for mammals. It just wants mammals. It doesn't care what kind of mammal. I think echidnas are out of the question, and platypus, but all the other mammals that we've looked at, and there's a huge checklist of animals out there that become infected with trichinella. It's not always the same species, but it's certainly the same genera. The tapeworms have huge ranges. Christina, I mean, lots of fish parasites and birds. I think every phylum of vertebrata has tons of tapeworm infections. They don't do much harm to the definitive host. And you're right. I mean, the intermediate hosts get nailed. And that's a problem for the host, not the parasite. So we did some experiments to see how long the worm could live in a dead host. We infected mice, we allowed the infection to go to completion, and then we sacrificed the mouse and left them inside of a fume hood for a couple of weeks, and then took them out and saw that the worms were still alive 2 weeks after the death of the host. So that has a survival instinct, a survival characteristic that favors its transmission as well. And I'm just wondering, manipulating the infected and uninfected ants, what does that do to the colony? I would love to know the answer to that.
Because I'll do your literature research.
Maybe I'll go back in the lab. Maybe I'll go back in the lab. No, people, no, please don't let that man back in the lab. And I can think of lots of experiments that you could do like this. Lots of chemical experiments to show what is altruistic behavior in an ant, right?
And they may well have done more research because this is a 2021 paper.
Right. So they were from Wiesbaden, right? Wiesbaden?
Mainz, I think.
Mainz and Wiesbaden.
Mainz and...
I'm wondering where Holdobler is from.
Yeah. I have no idea.
We can find out.
So they were from Cologne. They were from Köln and Mainz.
Right.
It is interesting, though, in our sort of human-centered focus, just to see that part of the dynamic here was caregiving, right?
Right.
There are these nurses, nurse ants that are actually taking care. And these infected ants, they don't need to beg. They don't need to say, hey, they're actually just preemptively taking care of them.
And better care than the queen.
Yeah.
And better care than the queen.
Pretty interesting. So.
Well, they can't chop, though. The queen can't chop. It's not like a human
the
queen.
point is, when a portion of the colony gets sick, there's a price to pay for the entire colony by food gathering and protection against predation and invasion by other species of ants that would like to take them over or kill them off, that sort of thing. So I don't think it's all positive here. To be honest, it sort of sticks the colony in one place. You can't move now. You're stuck taking care of these sick ants. And as a result, you're easy pickings for the woodpecker.
But I guess that's tough. Are they really sick, right? They're infected. But I'm not sure they're sick.
Well, they're lethargic. I thought you said they were lethargic.
No, they're not lethargic. They're just kind of more reverting to being like young ants.
Yeah, they're young. They're staying home. Everyone's feeding them. They're
watching a little TV.
The metabolic.
Watching the babies grow up.
The metabolism
watching their babies
measurements
grow up.
were fine. So they're not. And I think this is always good for us to kind of harp on the vocabulary. Like infected doesn't necessarily mean sick. You can be infected. You can be parasitized.
So we're all infected, and we're not always sick.
Yeah, we're always all kinds of things.
That's right. No, that's absolutely right.
We have viruses, bacteria, fungi. We have protozoa in us, right?
Yeah. So this might be a beneficial parasitome.
Parasitome.
Does a colony of sick ants take care of itself?
Oh, when I did that study, on the microbiome, on my own microbiome, they were actually looking for one protozoan parasite. They were looking for blastocystis. But that's the only parasite in the potential parasitome that they looked at. I don't seem to have any.
Good. That's good. Good news. Glad to hear it. All right. Thank you, Christina.
You're welcome.
That was exciting. That was exciting.
All right. Maybe this is not as exciting then.
No, it is. It is really. It's
great, Vincent. We're ready.
It's a really good paper, and it's a tough paper.
Well, it's actually the beginning. It's a candidate. It's Nature Medicine, a candidate antibody drug for prevention of malaria. And this is from many places throughout the U.S. And the first author is Catherine Williams, and the last is Daniel Emmerling. And of all the cases of malaria, 77% of them are in kids under 5 years. And the vaccines that we've talked about, at least one, RTS, AS01, is only 45% efficacious in kids after the first dose and goes down to 36% over 4 years. That's that RTS. There is a second one, R21, but they're both based on circumsporozoites, so I don't know if they're going to be any different. So there's a need for something else, and that could be monoclonal prophylaxis, right? And WHO has called for some other things for kids.
Vincent, I was gonna clarify that's deaths, right? Because we see a lot of malaria cases.
Yes, deaths.
But it's
correct.
the deaths that are clustered in the little.
That's
the little
correct.
Guys.
So in 2021, 619,000 deaths, and 77% of them in kids under 5 years of age. Now, monoclonals can be useful, but they're not so straightforward, right? They can be very expensive, and they note here that if they're too viscous, you have trouble delivering them. You need needles, of course. You have to manufacture them. So
that is difficult.
It's easy. It may be easy to find an antibody that works, but it has to have other properties as well. And so that's something that they also look at here. Viscosity, protein-protein interactions, aggregation, conformational and colloidal stability. And so there have been other monoclonals I published that can prevent malaria. These have been tried in humans. For example, one has 88% efficacy, prevention of infection actually for 6 months in endemic regions. Two others have been tested in clinical trials. But again, these are not so great for production. So they wanted to identify some monoclonals here that might be better produced. So they actually report over 50 preclinically protective, that means in an animal model, And these are taken from people who were immunized with the RTS-AS01 vaccine. Interesting. And they got plasmablasts from these patients. And so they have, they take PBMCs from these vaccinees who were in the clinical trial for RTS-AS01, and they got 3 doses ultimately. And they also got challenged with malaria and controlled human infections. And so, and then some of these patients also got a 4th dose of vaccine and got challenged a 2nd time with malaria. So you have all kinds of, you have people multiply vaccinated, infected twice. So they collect plasmablasts from these patients, and they determined the genome sequences to generate paired heavy and light chain IgG sequences from these plasmablasts. And so that's going to be the basis for the monoclonal. They're going to start with the IgG heavy and light chain sequences. And they do a lot of analysis of these sequences. And they have, for example, they could group the immunoglobulin genes into lineages. And they notice that sometimes they have lineages in multiple plasmablasts, which they say indicates recent cellular expansion in lymphoid organs. And so they think that's good. So they focus on these expanded lineages. And they say the lineages with the largest number of plasmablasts per vaccinee. So in other words, the same sequence in different plasmablasts, they thought were the ones that they want, because they probably have won the race. They've outcompeted other lineages for antigen binding and so forth in the lymphoid organs during the selection process. So they basically took these sequences and they decided which ones they wanted by criteria, including the ones that I've just told you, and they produce them in cells. They introduce them in cells and make antibodies, proteins that are purified. So they basically establish cells that are producing these antibodies. They did 369 clones. Each was a unique plasmablast lineage from these patients, again, who have been immunized and infected. And they're screened by ELISA, and 38% of these monoclonals, 139 out of 369, reacted with circumsporozoite. And those were, of course, moved further into the paper here. Next, they want to look at protection of mice. They select 77 of these monoclonals for looking at mice, and they have mice infected with Plasmodium berghei, and these are actually transgenic sporozoites that express Plasmodium falciparum CSP rather than the berghei CSP, because these are human monoclonals against human. They're against PF. CSP. And a third of the antibodies were from unprotected vaccinees, people who got malaria, and half of those showed near complete protection in mice. Over half of these antibodies provided over 95% inhibition of sporozoite liver burden in mice that were infected. So they conclude that antibody-mediated protection after you get vaccinated with RTS vaccine is not just based on antibodies, but also probably affinity maturation, other T cells in the blood as well. So it's not just the antibodies that are induced by the vaccine. They looked at what these antibodies are binding by various techniques like plasma resonance, so you can measure the affinity of these antibodies. And the ones that they selected for further study had a broad range of affinities against circumsporozoite protein. They say that maturation of these inhibitory antibodies to these short homologous peptide sequences that they're binding to, they do some studies with peptides derived from CSP, is probably benefited binding to heterologous peptides as well. They take 70 antibodies and they evaluate those. Yeah, go ahead. Yeah, did you have a question?
No, I just remarkable that. No, please go on. I was just
they take
reacting to the data.
They pick 70 antibodies and they compare them in a sporozoite challenge mouse model, and they're comparing it to a monoclonal that had been previously published but wasn't used clinically because it was too difficult to produce. Because they say we want to prioritize which of these to develop because you can't do all of them, obviously. About half of them demonstrated inhibition in this mouse model comparable to this 317 antibody that's been published before. And one was even better. 224 had superior activity in this mouse model, this mouse challenge model. So they took that antibody for further advancement. They produced it and characterized it. They actually have over 30 antibodies with comparable activity to 317, which could be brought to clinical development. So that's the protective ability of these antibodies, right? So then they say, for clinical development, we want to we'll look at other properties of the antibodies, pharmacological and biophysical characterization that would be needed for successful production. And they looked at 26 monoclonals, essentially, made sure that they had maximal activity in mice in the sporozoite challenge model. And then they looked at manufacturability, stability, and formulation components, for example. They looked at aggregation, biophysical stability, and lack of viscosity, and they pick one antibody, 7088. So it had comparable neutralization or protection ability as the other ones, but had superiority in terms of these biophysical properties. I think that would be easily brought to clinic and manufactured, in fact. Okay, so they have one monoclonal they want to use. So then they further engineer the antibodies in the framework regions just to make it even better to be produced in the manufacturing process. And they introduced amino acid changes. So these are in the framework regions of the antibody, which don't affect antigen binding. But these have been shown by others to improve developability. In other words, these properties of the monoclonal that are necessary for manufacturing and being given to humans. And they introduce these changes, and then they have to check each of the antibodies that they've changed amino acids in, and they retain monoclonal antigen binding profiles of the parent. So these changes are not affecting antigen combination. Then they also introduce an amino acid change into the Fc domain of these antibodies that extends the half-life of the antibody. So this is a known amino acid change that will extend the half-life. It's called Xtend, Xtend. And, they say because we have low serum antibodies in the mice, and so maybe this would extend the half-life and make it last longer and so forth. So they make these variants. They put them in cells and produce them. And then they go through many, many more clones of this one, and they identify the best producing cell line that makes, in bioreactors, that makes the highest levels. And they identify, they go through some of their other assays, and they identify a monoclonal, which they call MOM01, which looks like it's going to be great for production. It has the right binding properties to CS, circumsporozoite protein, and it did great in the mouse protection assay. So they say this material is being used to support studies for clinical development of an anti-malaria drug. So this is in a clinical trial, NCT 05891236, and presumably that'll be another paper that we'll see at some time in the future. So in a remarkable amount of work going into this. But they also learn along the way some interesting things, which I want to just review. First of all, there's an inverse relationship between the percentage of circumsporozoite producing or circumsporozoite specific IgG antibodies and vaccinee protection status in the original clinical trial, right? Which they say suggests that more cells making anti-CSP or anti-other residues may not drive stronger protection. It's something else that's driving the stronger protection. And secondly, they say sporozoite inhibitory activity in mice doesn't correlate with binding kinetics to one of these CS peptides that they look at, but it does look like it looks like the off rate corresponds to some of these peptides that they've used. And so again, protective antibodies that are induced following the vaccination with this RTSS vaccine probably affinity mature to these short peptide repeats that they've been looking at. It's really interesting. And finally, they decide they make a hypothesis that multiple peptide repeats act as an immune decoy that dilutes protective immunity. So if you have binding to another antigen, not CSP, it dilutes protective immunity. So you want to make sure that your antibodies are binding to the right peptides. So you have the interesting findings from this development, but also a candidate that looks like it has good properties, and hopefully in people it will show some efficacy.
Yeah. So I have a question about the amount of antibody per sporozoite that you need to neutralize the sporozoite. How many antibody molecules need to attach to the sporozoite before it's not affected, before it's not infectious?
Mm-hmm.
It's a good question because you remember the vaccine that succeeded out of Oxford didn't succeed until they saturated the artificial carrier for the peptide. And actually, they used the entire protein. Until they could saturate the surface of this, let's just call it a carrier.
I guess I'm going to turn that around. I'm going to turn that around because I don't think the problem with the vaccine was getting enough antibodies on the on the actual sporozoite. It was getting enough antigen stimulation for the immune system to trigger a high enough serum antibody level.
Yeah. That's right.
Because what they're trying to do is get so much antigen that you can get a really good. So, but no, it's the right question because the question comes is how much of this antibody do you have to give?
Right? Exactly.
Because that's your cost issue.
Well, there's another issue here too, and that is that each person receiving this, it's not going to last forever. There's going to be a half-life of the injection dose. And let's say that you've got an overabundance of antibody molecules to begin with in the injection dose. But 6 months from now, how much would be left to protect against another outbreak that might have occurred within 6 months? So they could have answered that with the mice experiment, but they didn't, unfortunately. Because this is going to be a clumsy system, don't you think?
Well, so it depends.
Every 6 months, you're going to have to give more antibody to get the levels back up again to where that was effective.
What do you think of that, Daniel?
So a couple things here. So I'm trying to run the numbers. So I was thinking about what were the doses that we had to give with the monoclonal antibodies for for COVID, right? For SARS-CoV-2, right? So we'll take bebtelovimab, 175 milligrams, that was the dose you got. So I'm going to round it to 200. I'm going to be sloppy. So how much does it cost in general per gram of monoclonals to make, the manufacturing, not the research costs? So it's about $100 a gram. So that means you can divide that by 5. So about $20 for 200 milligrams. It's a lot of money. They're telling us that they might be coming up a way to get the costs down. So we can get the costs, like where's that price point going to be? The other thing which they do talk to us about, so not only can you get the cost down, but by modifying the FC, we're getting better and better at adjusting the half-life of the monoclonals, right? So we'll go back 20 years to when we developed denosumab, Prolia, Xgeva, right? We engineered that for a 6-month half-life. You give it every 6 months.
Right.
So, here comes kind of the question, what dose do you have to give? And in a lot of areas, malaria is seasonal, right? Much like we think of flu and RSV. So do you give the dose in, think of it as our, season, like let's say the season's going to be November, December, January, you give this dose in October or September. So let's say you get the dose down to like $5 a dose, but with that $5, you're not going to get a 35% like the Oxford. You're going to get an 80, 90%. So what is sort of the break point? And a lot of this money is going to come right from Gates and other foundations. So what is the price point? What is a child under 5's life worth? What are we willing to pay? How much money can we get?
Question.
Well, no, but that is the question.
I know. I do know this.
Is it $5? Is it $10, right? We're talking about over 500,000 little kids under 5 dying every year. That's the state of where we live right now. And if we can get this down to a low enough cost, if we can get a long enough duration, particularly in areas where there's a seasonal, it may not have to be given twice a year. You may be able to get in there with once a year. I mean, I see this as being, really exciting work. And because it's one thing, as we're seeing in this, that you give a whole bunch of vaccine and the level of those antibodies you generate doesn't necessarily correlate, as they point, with immunity. It's the level of the right antibodies. So instead of sort of rolling the dice and hoping your vaccinee makes the right antibodies, let's just give them the right antibodies.
Unless you can figure out how to get the right antibodies induced.
So this is the right antibody to prevent infection.
Right.
This is not an intervention to somebody who's already sick from malaria and needs his life saved. You wouldn't give this antibody to those people because it doesn't react with any other stage except the invading stage.
Yeah, it's not therapeutic.
The COVID story is a little bit different because you've got a virus particle that's not going to be different, and you can knock out X number, then you're below the clinical horizon for that. But for malaria, it's a trickier slope.
This is like Evusheld, right? This would be like the stuff you get ahead of time. It drops your risk of even getting sick by. So it's that.
You know who's going to really love this?
Malaria shield.
How often did you have to give Evusheld, Daniel?
It was every 6 months.
6 months. Okay.
Yeah.
I can tell you who's going to love this drug if it works.
The military.
And that is the military. That's exactly right. Exactly. And doctors.
And
working
travelers,
in endemic areas.
Could be traveler.
And travelers. This is exactly. So do you think this could be engineered?
Individuals?
Could this be engineered into a pill that you would take orally?
So that's the problem, right? It's an antibody, right? And antibodies. It is interesting. Like, I face that with a little bit of negativity, but let's think about it. Like when you breastfeed, right? We always talk about the baby.
Of course, that's what I'm thinking. That's exactly what I'm thinking.
Yeah.
So you've got 6-month-old kids. What is the major peak of susceptibility after the maternal antibody wears off? It's 1.5 to 2 years, maybe 3 years.
Yeah. So we may be able to.
You could probably give them a pill, or even in their formula, and get them protected. They can still absorb antibody.
Yeah.
The problem is that they're not getting any more because the mother's antibodies have waned. Yep. Her breast milk doesn't contain the same level as it did when she was first delivering. And you've got these other kinetics going on that confound all this, right?
And I'm not sure the needle is such as an impasse, right? Because as we know right now, we're rolling out malaria vaccines throughout Africa. We do a lot of vaccines injectable. I mean, so
but vaccines are different.
There are networks in place. Think about your,
they're
your
different.
MMR,
I would argue that what you're dealing with here is not a vaccine, all right? It's a treatment.
It's
a preventative treatment.
Yeah, I guess it depends which audience you're talking to, right? It's a passive.
But it's not going to be lasting. It's not going to last like a vaccine. It's going to wane like a drug.
Yeah, but as we know with the antibody levels in our vaccines, if the antibody is what's critical, right? Nirsevimab, right? That's Beyfortus. That's on the vaccine schedule, right? That's a passive vaccination. And I think the challenge is that certain things like malaria, you may need to maintain a high antibody level for the protection. By the time T cells kick in, by the time the antibodies come back up, the little kid under 5, it's a little too late. Sporozoites.
So what about
the liver.
The analogy using COVID and the circumsporozoite protein and looking at an RNA vaccine?
I think we looked at. Did we not discuss a paper?
No, it's being looked at, right? I think
we did. Yeah. I'm sure Vincent talked about an RNA vaccine.
We did.
Did you not?
Yeah,
you did. Yeah. So that's on the go.
Yep.
To just have the right antigen now, that's the problem.
Yeah.
Problem is the right
I think
antigen.
we just need to see this in the context of the broader efforts to control malaria, and it's going to be another tool that will be used in combination with vaccination and insect netting and residual spraying. So I think it's just going to be an additional tool that maybe will edge us a little bit closer to having a good handle over malaria.
But I like it if we can get this to be less expensive.
I do like this.
And maybe, and we talk about needles, but maybe those micro patches that they're working on. And we talked to some people at ASTM about that. You just put a little sticker on your arm and it's got all these tiny little needles in there. And now you've got your...
Great. Continuous flow.
And actually, sometimes it's more difficult to get a young child to eat a pill than to give a quick injection.
Or to do anything for that matter.
All right.
Yeah, that's wow.
That's TWiP.
That's a really, really good paper, Vincent.
Thank you. No problem. TWiP 228. Show notes, microbe.tv/TWiP. If you have questions or comments, you can send them to twip@microbe.tv. And if you like our work here, please support us, microbe.tv/contribute. Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Oh, thank you. And everyone, be safe.
Dixon Despommier, trichinella.org, thelivingriver.org. Thank you, Dixon.
You're welcome, Vincent. And by the way, your reference to Yul Brynner was for trichinella, not for cysticercosis. So we can talk about it some other time. I would love to come back to that story.
That was Daniel who said that, yeah. Christina Nowell is at the University of Glasgow. Thank you, Christina.
Well, thank you. I had a really good time.
Great paper.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back in 2 weeks.
Another TWiP is parasitic.